Thanks.
Okay, hello. Hello, welcome to our distinguished lecture series. It's my great pleasure to introduce Condon. We first met when we were in a group of grad students and postdocs visiting the Math Sciences Research Institute in Berkeley in the mid-'80s. And that's up on a hill, so the challenge was to bike up and down that hill. And I remember there was a researcher from Holland, which isn't known for its hills, who had a very disastrous crash landing coming down that hill. Do you remember the lane? Anyway, but Anne, coming from Ireland, was very good at that bike ride, I think. She was at that time a graduate student at the University of Washington. She graduated in '88 and won the ACM Dissertation Award for her thesis, and went on to a faculty position at the University of Wisconsin, all of the W's, where she had an NSF National Young Investigator Award. She was there for almost 10 years, I think, and for the last 9 or so has been at the University of British Columbia, where she holds an NSERC General Motors Canada Chair for Women in Science and Engineering, and is Associate Dean for Faculty Affairs and Strategic Initiatives. She's done a lot to recruit and retain women in computer science, including being very active with the Grace Hopper Celebration of Women in Computer Science, which she chaired this year. And amongst all of that, she's very active in research in a number of areas, algorithms and complexity theory, and bioinformatics, which is what she will talk to us about today.
Thanks, Anna. And thanks very much for the invitation to speak here today. I'm going to talk about DNA and RNA, which are two molecules that have fascinated me for the last 13 or 14 years or so. Actually, I think it was in about 1994, which I was just commenting a couple of times earlier today was a very good year for lots of reasons, that Len Adelman published a paper in which he described how one could compute with DNA molecules. And I think that paper was something that inspired a lot of people to think in new ways about DNA and about RNA, including myself. And sort of in the process of doing that, I got interested not only in designing molecules to do new and interesting things, but also in predicting what molecules do sort of naturally, in particular what kinds of structures they form as a function of their sequence. So I'd like to start today by just telling you a little bit about DNA and RNA. I presume there are some people who don't know a whole lot about these molecules and the structures they form. And some background about algorithms for predicting structure that I'm interested in particular secondary structure. And this piece of, the work I'll describe there is work where I've made some contributions over the years in secondary structure prediction. And then in the second half of the talk, I'll step back a little bit and talk more broadly about what I think are interesting directions in programming molecules or designing molecules to do new things. And the examples that I'll provide are not examples that I'm working on myself, but they're examples I do find very inspiring and I hope will be stimulating for you to hear about. So hopefully everybody is familiar with the DNA double helix. The double helix forms because of base pairs between complementary molecules on two strands. So in particular, A likes to bind with T and G likes to bind with C. As you can see in this picture, these base pairs cause the molecule to form an interesting structure, which we call the double helix, which has a right-handed twist, which actually is also the same structure formed by rotini pasta, in case you ever need a check. It also has a right-handed twist. And since I'm someone with very little geometric intuition, I would have thought if you turned one of those upside down, it would have a left-handed twist. But it doesn't, amazingly enough. It's always a right-handed twist. So I'm starting to get a little bit of geometric intuition here. Anyway, my interest is not so much in the 3D or tertiary structure of these molecules, but in the secondary structure. And the secondary structure simply tells you which bases are pairing with which bases, which is extremely boring in the case of a DNA double helix, because the first one pairs with the first one, and the second one pairs with the second one, and so on, up both sides of the helix. But if you took a DNA double helix and you separated the two strands from each other, and you put one of them in a test tube or in solution, it wouldn't have its complementary partner to pair with. So it would have to fold into something different. And basically, the same principle applies. Beads on the sequence, A, C, Gs, and Ts, look for each other. A's are looking for T's, C's are looking for G's, and they will bind up when they find each other. Okay? That's a very simplistic explanation, but that's roughly what happens. Okay? Now, if they're matching sequences all over the strand, you can, as a result, get interesting structures like this branch structure here, which has an outer stem at the top and then 3 branches coming out underneath. So a tree-like structure. And these kinds of structures are very common in biological RNA and elsewhere. This particular one is very reminiscent of something called a transfer RNA, which is extremely important in translation of RNA to proteins. Again, I want to caution that if you were to look at the 3D structure of something like this, it would look totally different. It wouldn't look anything like this cloverleaf structure. Rather, parts of it have completely moved towards each other and bonded. So remember that these secondary structure drawings are extremely simplistic. They tell you really very little about the 3D structure. And the secondary structure, which is, again, just the list of bases that are pairing with each other, is purely sort of a combinatorial object. So keep that in mind. You can also get structures forming which are not branch structures. So here was a structure that maybe originally had 2 branches, and then these 2 looped parts at the end of the branches came together because they were matching base pairs for those to form. This kind of structure is less common, in particular because if you keep in mind that these helices here have a twist, well, also if multiple base pairs form between 2 loops, they have to twist, and obviously eventually there are going to be sort of natural constraints that will impede too many of those base pairs forming. But it can happen. Here's an example of a structure that was designed. This is not a naturally occurring structure. It's designed to be built out of DNA. I chose this example in part because it's designed, and in part because it's actually made up of multiple molecules, and the previous examples were all single molecules that had folded. Here, the different colors represent different molecules, and they were carefully designed to weave together in a very interesting way. And there are 4 pieces sticking out at the ends that have the potential to bind with other things in interesting ways. This particular structure is designed the way it is because it's quite rigid, and the rigidity is sort of important in its application, which I'll describe later. Otherwise, it could be quite floppy, which wouldn't be so great for the particular application. The next example I'll describe is also examples of multiple interacting RNAs that are forming interesting structures. And in this case, it's sort of a pathway of structures that are formed that's interesting as opposed to some kind of static end structure, as we saw in the last example. So here, imagine that we have 2 simple structures. I didn't put in all the base pairs, but imagine that there are base pairs forming all the way up here. So you've got a helix or a stem with a little hairpin loop up at the top. And they're in a fairly stable state because many bases have found partners to pair with all the way up these stems, although there are unpaired bases up at the top. Now, if a 3rd molecule is introduced, an initiator molecule, we'll call it, and it happens to pair up nicely with the end here of this one. This would be called a toehold, and this can get latched on here on the toehold, and then it can actually disrupt the base pairings all the way up the stem and pair with this one, causing this hairpin to break and this top part to be free. And so the result is something like this, where now the initiator molecule is paired with the left hairpin. The top part of it is free, and the other molecule is still here. And you may notice now that this toehold could actually pair with the top there, causing this molecule to escape from sort of a metastable state into a state where it also unfolds. And then if you look really carefully, you might notice that the part that's freed up in this molecule would actually then pair with the first one again, and you could get a cascading sequence of reactions if there were many, many copies of these molecules in a pot, producing a really, really long double-stranded chain of these hairpins. And this was designed for an application called molecular detection. If there were tiny quantities of the initial initiator DNA in a cellular environment or in a tissue sample or something, and it triggered this cascading chain reaction, the product would be a very long, interesting DNA that could be detected. And in fact, the length of it would tell you something about the multiplicity of initiator molecules in the sequence. So it's an example where the pathway of structures that is formed from the molecules has been designed in an interesting way and can be used for molecular detection. Okay, so the questions, that I'm interested in, motivated by these examples, are first of all, given one sequence or given more than one sequence, can you predict the structure that it'll form, just the set of base pairs that form when the molecule folds, or the pathway of structures that it would go through in the process of changing shape? And second of all, could you program sequences, could you design sequences that would form a particular structure or follow a particular pathway for some reason, such as the last example where the application would be molecular detection. I'm going to spend most of the talk on the prediction aspect, and then at the end I'll come back to the programming aspect a little bit. So if you think for a minute about a long, say, DNA sequence or RNA sequence, it's just a sequence of A, C, Gs, and Us. Well, if there's plenty of each of those in the sequence, there's going to be an exponential number of ways that A's could find T's and G's could find U's. So there are lots of different secondary structures that the molecule could fold into. So how are we going to figure out which one it actually does fold into? Well, in order to do that, you kind of need to understand something about the principles of structure formation. And I like to think of it as a probabilistic process that's driven basically by thermodynamics. That simply means that the molecule is like a couch potato. It's lazy. It wants to be in a low energy state. And in order to do that, base pairs release energy, so it likes to form lots of base pairs. So let me just click on this. I should have checked this. Look at that, it works. So you can see this molecule randomly forming base pairs, breaking base pairs, and so on until it finds this sort of stable fold in which it's pretty stable. And of course, in a real molecule, in a real test tube, it's not going to do this and then stop. There's a constant forming and breaking of base pairs. So the right way to think about it is that the true structure is somehow the most likely state for it to be in, or the state you're most likely to see if you were to look into the test tube and look at the state. Okay. So if you believe in this couch potato model, what you want to do is find the lowest energy state or the lowest energy secondary structure for this particular molecule. And if your thinking is a little more sophisticated, you would be interested in thinking about it more probabilistically and asking what are the probability of individual base pairings that form when the structure folds, because it isn't in the end a static thing. And there's a quantity called the partition function which can be calculated to help you figure that out. I'm really not going to talk much about the second problem, but I will just say that the first one is conceptually simpler, easier to talk about in a presentation like this, but all the methods for the first one apply very simply to the second one, so keep that in mind. So what I'm saying is also useful for the second problem. Okay, so if the goal is to find the minimum energy configuration, we need to know something about the energies of structures, like how to score them. What is the energy of a particular structure? A simple model simply says, well, base pairs are good. They stabilize the structure. They reduce the free energy. And so we'll give each base pair a score, and we'll simply sum the scores to get the total energy. So that's a reasonable working model, but it's a bit simplistic. It's not what's used in practice. What's used in practice is called a loop-based energy model, where instead of associating scores with individual base pairs, we associate them with loops. So at the end of a stem here, we've got something called a hairpin loop, which is defined by the unpaired bases around the edge here. There may be many, and the closing base pair here. A simple consecutive pair of base pairs would also be considered a very simple loop here called a stacked pair. And then some loops can be quite complicated, having multiple branches, and those are called multi-loops. So there's a few categories of loops, and biochemists have spent a lot of time figuring out what would be a good way to score each particular loop. In some cases, for example, stacked pairs, for every possibility for the 4 bases that could appear here, they will have a very precise energy score. And in other cases, like multi-loops, they have less information to inform their models, but they have some estimates. Again, it's a very simple sum the scores to get the total energy once you have the contributions for the individual loops. So a good starting point for a computational person would be to go to your favorite biochemist. Doug Turner at the University of Rochester is a great one, and say, can you give me a table of energy values? And then you can write a function that will calculate the energy of a particular structure, and then you're left with a nice clean optimization problem. And just a little bit of notation that I'll use later is to refer to the energy of a sequence X folded into a structure Y, given energy values theta. This would be a vector of values that say this is the way to score this multi-loop, this is the way to score this hairpin loop, this is the way to score this stacked pair. That energy would be denoted by delta G of X, Y, and theta. Okay. So unfortunately, the general problem then, given a sequence of figuring out what is the lowest energy secondary structure, the problem in its full generality is NP hard for even a simple energy model, loop-based energy model. So that's kind of too bad. But in practice, it actually isn't that important because most structures we see or we're interested in are not remotely that difficult to fold and to find the minimum free energy structure for. So the way I like to think about the algorithms that are out there for this problem is to look at their efficiency. So these are algorithms that are truly finding the minimum free energy. They use dynamic programming. They're not heuristics. They're not approximating the free energy. And so we can ask what's their efficiency, and also what kinds of structures can they handle. They can't predict all possible structures because otherwise they will be solving an NP-hard problem. But for example, all the structures that just have branches, which would be called pseudo-not-free, can be predicted efficiently, and so on. So the generality axis is a little tricky to define. But before I do that, let me just also say that as I describe a little bit about the algorithms, I need to tell you a little bit. I'm just going to change the way I draw structures for convenience. So I used to draw them like this, but another way you could represent exactly the same information is like this. So now the sequence is organized along the bottom from left to right, and base pairs are drawn as arcs. Okay, so this particular one here at the closing base pair of an internal loop is here, and the others are out like that. And so this is just a change of representation. Here's my cloverleaf structure and how it would look in this arc diagram representation. Both of these are examples of what we call pseudo-not-free structures. They're branched. That's all it means. They look like a tree. Here's my structure that's pseudo-knotted, where 2 loops came together. It's called a kissing hairpin structure. And you can tell immediately when something's a kissing hairpin structure if you look at its arc diagram, because there are going to be crossing base pairs. So people distinguish very much between pseudo-not-free and pseudo-knotted. And then among the pseudo-knotted ones, this would be called a kissing hairpin because 2 hairpins are coming together. And a simpler type of motif would be called an H-type pseudoknot. And in that case, one of these would be gone. You would simply have a hairpin and then the strand looping around and binding to it, but there wouldn't be this 3rd stem here. Okay? So in terms of generality, I'm interested in branched or pseudo-not-free structures, this simple type of structure with 2 interleaving stems, which would be called an H-type pseudoknot, and then this kissing hairpin type structure here. So I could label my generality axis with those types of structures. So here in a nutshell is how you would predict pseudo-not-free structures, and these algorithms have been around since the late 70s. They're simple and beautiful algorithms that use dynamic programming. And I think of this algorithm as sort of a peeling and chopping algorithm. If you've got a pseudo-not-free structure, you can sort of reduce it to nothing by a sequence of either peeling off base pairs from the outside in or chopping it into 2 independent parts, which is what you do if you were in this region here. So you can always describe a structure in terms of smaller structures by either peeling or chopping, basically. And so you can build a dynamic program or algorithm around that, which is going to keep track of minimum free energy structures for all subsequences of the full sequence. And it knows that it can compute the minimum free energy for a particular subsequence by either peeling or chopping that particular subsequence. Okay? So on the left here, I have an abstract representation of an unknown structure for a particular subsequence of the whole sequence. And I need to minimize over all ways I could chop it or I could peel it, and then I'd be left with a substructure. And I can just take the minimum over all those possibilities, and I'll figure out what the minimum free energy structure is. So that's sort of, in a nutshell, what the algorithm does. Very natural and simple. When you get to pseudoknots, it gets a little more complicated because you've got these crossing base pairs. So if you look at this overall structure, you can't chop it into two independent parts. And if what you've got to work with is the full structure, you can't sort of peel from the outside in to get smaller structures because the thing at the start isn't paired with the thing at the end. Okay? So you need an additional step or alternative to peeling and chopping, which I call a banana split, because you're basically splitting the thing into two parts that kind of look like a banana shape. Okay? And that's going to be bad for the complexity of your algorithm, because now you're going to need to be able to keep track of all subpieces of your overall sequence, where you define the subpiece by 4 components instead of 2 components. Okay? So you're going to need n to the 4th space to sort of store minimum energy structures for each of these bananas. Okay? And then it's not at all clear how you're going to further develop the recurrence for each of your bananas right into to further things. So one approach people have taken is to say, okay, well, if we've got one of these pieces, we could peel, and that would be a nice, easy thing to do that wouldn't increase our complexity by too much. So we could use a peeling mechanism on our subpieces. And that will give you an n to the 5th algorithm with n to the 4th space that can handle situations where you only have 2 of these overlapping pieces, because you could break your 2 pieces into 2 parts that are bananas, and then you could peel away on the bananas to get to solve your problem. But it doesn't quite work when you've got 3 overlapping parts. And it's actually, this is a biologically interesting motif. You do find kissing hairpins in biological structures. So it's interesting to ask if you could do that in n to the 5th. And the earliest algorithm that could do these kinds of structures was due to Rivas and Eddy, and it was an n to the 6th algorithm. It could actually do many beautiful structures that were way more complicated than this, but it needed n to the 6th time. So one of the contributions of one of my students, Hosna Jabari, and also a colleague at Caltech, Ho-Lin Chen, who's a postdoc there, and myself, was to figure out how to extend this peeling idea to handle structures like this in n to the 5th time. And our idea is extremely simple, actually. I can't believe it took us this long to think of it, or anybody else. And that is that if you've got one of these banana splits, you can always peel from the outside in. But in fact, you've got sort of 4 points defining one of these bananas. And there's no reason why you actually can't peel away on just one side, or peel away on the other side, or peel from the inside out. And if you can alternate between all those different ways of peeling, you can do much more general structures than you can do with the original n to the 5th algorithm, which could just do 2 of these. So I don't want to go into the details of the algorithm. I mean, the main message is that, hopefully I've got it on this slide, that there's been a lot of interest in the efficiency of finding minimum free energy structures for different types of pseudoknot-free and pseudoknotted structures. And the complexity ranges from n cubed for pseudoknot-free up to n to the 6th for extremely general structures. And kissing hairpins and even a little more general can be done in n to the 5th, which I think pretty much covers many of the structures you're likely to find in biology or even in design work at this point. So I think it's a good complexity.
Sorry, what is n?
N would be the length of the sequence. Sorry, I should have said that. Yeah. So in practice, the n-cubed algorithm works very well. I mean, biologists can use it for all practical purposes. for pseudoknot-free structures. But if they want to do pseudoknots, n to the 5th time into the 4th space is pretty bad. So I think there are more tricks you could apply that would make the work somewhat less elegant, but quite practical, while still guaranteeing minimum energy for a slightly more inelegant set of structures, but could be very useful in practice.
But what is it? How large are the problems in practice? And is there any great concern about ones that are 10 times as large or 100 times as large as the current ones?
So in practice, people are interested in anything from 20 or 30 nucleotides to a few thousand nucleotides. And you can do a few thousand in n cubed time, n squared space, no problem. But you will run into trouble with the pseudoknotted ones at that. But you can do a few hundred easily with a desktop machine. After that, you're starting to run into trouble. Okay. However, the bigger problem is the accuracy of these algorithms. So when I started to think about them, I was all interested in, could you do it in n to the 5th? Could you do it in n to the 4th? But in practice, probably nobody's going to use your algorithm anyway, because it's probably just not going to give you a very good prediction, no matter how fast it is. So that's kind of a bigger problem that I sort of slowly came to appreciate. So I now want to turn to the third axis in my diagram, which is accuracy. So I said nothing so far about accuracy. What I've said is that we have algorithms that are guaranteed to find the minimum energy structure. So any problem with these algorithms has nothing to do with their heuristics or their approximations or anything like that. So if they're not producing perfect predictions, it must be because their energy model is wrong somehow. Okay? And we can measure the accuracy. The one way that I'm going to use today to measure the accuracy of the algorithms is something I call F-measure, which is simply the harmonic mean of sensitivity and positive predicted value. And sensitivity is sort of measuring how many base pairs did you get correct out of the true base pairs in the structure. And positive predicted value is measuring how many of the bases you predicted, how many are correctly predicted. So taking the harmonic mean of these is good because it sort of implies you need to find a balance between over-predicting things that might not be in the structure or under-predicting things because you're too cautious. You have to balance those two things. And so the accuracy is going to be some number between 0 and 1. And if you get accuracy of 1, you've got perfect prediction. And if you've got, or 0 and 100%, if you've got accuracy of 0, you've got nothing right in the prediction. Okay. So if you take the standard algorithms for pseudoknot-free structures and you use the state-of-the-art energy model, which is this Turner model, which is developed by the Turner Lab at the University of Rochester, and you run it on a huge database of structures, an average over the whole lot, which my student Mirella Andronescu did, you find that you're getting about 60% accuracy, which is pretty darn good, say, compared to what you could do with proteins, but leaves a lot of room for improvement. Okay, so at some point I started to get more interested in trying to make this number get better than trying to get more efficient algorithms up sort of on the other two axes. Unfortunately for me, so I thought this was a really interesting problem, but I didn't really think I knew how to deal with it. I'm not the kind of person that knows how to build large databases of structures and infer new parameters and all that. But luckily, one of my students was really interested in learning all about machine learning. So she took this on, and she's done an incredible job in sort of advancing the state of the art here. She just defended her thesis on Friday. So, okay, so I'd like to talk about this problem for a little bit. So the energy model of the Turner Lab has about 350 features. So a feature might be the stack pair CG-AT or something like, or AU because it's all RNA, right? Or it might be a hairpin with 4 nucleotides along the top and a particular closing base pair. So a feature is a loop, right? Because this is a sum of loops, okay? And each feature has a value. Some of these values have been experimentally determined. Wet lab techniques, optical melting temperature methods have been used to figure out the values fairly accurately. However, they can't do that for all the different features. It's very difficult. So others are simply fitted to known structures. There are large databases of known structures out there that we can work with. There's also, well, as I just said, a lot of available data. We've got structural data where we have a sequence and we know its structure. And we also have the data that was carefully curated and determined by the Turner lab in particular, but other labs too, where we not only have a sequence and a structure, but we also happen to know the energy associated with that particular structure. And I should have said earlier on, of course, the secondary structure depends on parameters like the environment, like temperature, pressure, and so on. And I'm sort of assuming standard conditions throughout. So that's why I can always refer to the structure. The structure would be different if you heated it up, but I'm assuming standard conditions. So we have a model and we have data. And what we'd like to do is learn the energy parameters better so that we could get better predictions with the model we have. So we'll keep the features fixed. We'd like to improve the parameters because we think that the ones that were fitted to known structures may be a bit unreliable. We think the ones that were experimentally determined are pretty reliable, but not the other ones. Okay. So we'd like to use the data we have, all the structures we know of, all the thermodynamic data where we also know the energies. We'd like to stick with the Turner model because that's what everybody uses, and if we're going to venture down this path, we don't want to start changing the model under the feet of the biologists. We kind of want to keep that fixed. And of course, we have an algorithm that we'll use as a subroutine, which we know will give us the minimum free energy prediction. Okay? And we're very careful to keep the thermodynamic data because, again, that's very reliable and trusted by the biologists. And what we want to do is figure out a vector of values for the energy values that will give us the best possible predictions. So that's the parameter estimation problem. And there's a very natural way to formulate that as a Boltzmann likelihood parameter estimation problem. And in fact, before we published work on this, it was already published by Doe et al. At Stanford, who proposed this type of approach. And basically, the idea is simply to find a vector of energy parameters that maximizes the likelihood of the structural data. So the probability of the structures given the parameters. And this is kind of why I mentioned earlier that we should think of structure formation as a probabilistic process. And when you do that, talking about the probabilities of the structures given the parameters makes some sense. You can formulate that in a natural way. And you can use your algorithm or variation of it to figure that out. What we did in our work, which Doe et al. Didn't do, was to extend the objective function not only to maximize this likelihood, but also to try to make sure that the energies of the structures in the thermodynamic data set are close to the measured energies. So we want to respect the thermodynamic data. And we added a regularizer to avoid overfitting. And in order to implement this, we actually needed a bit more than just these starting points, the energy model and the minimum free energy algorithm. You need, for example, to be able to calculate the gradient. And that required actually a lot of programming work to make that all work. So this was a lot of work to implement. And it's also very time-consuming to run. But you could get it to work. Mirella could get it to work. She also tried a different approach, constraint generation parameter estimation. And the idea here is to say, well, the true energy vector should be such that the energy ascribed to the true structure is lower than the energy ascribed to the wrong structures, right? That should be true. And what's more, the energies assigned to structures in our thermodynamic set should be close to the measured energies. So we'd like our parameter vector theta to satisfy these two constraints. So when you say constraint generation, these are our constraints. Okay? Okay, so in those constraints, there were these odd Greek things here. And those are just slack variables. We don't expect to be able to find a vector that will perfectly satisfy our constraints. So we introduce slack variables, and we ask then, can we minimize sort of the values of the slack variables? We want to bring those as close to zero as possible, such that we satisfy these constraints. Okay? If we could find such a vector theta, we'd be in good shape. Now the problem with this, one of the problems with this, is that there are going to be an exponential number of constraints here, right? Because there's an exponentially many possible alternative structures for the true structure. So Morella's idea was simply that we could generate constraints on the fly. We could start with our initial parameter set and generate a bunch of low-energy structures and require that the true structure would have energy lower than any of those, which it might not have because the energy vector we'd have might be wrong. And then as the algorithm, we'd run this algorithm, we'd get back a better, hopefully, parameter set, but it would still be wrong. And we'd use that to guide adding additional constraints into the program. And we'd keep repeating that until we got to a pretty good parameter set. And this turns out to be much easier to implement. You don't need any function to calculate gradients. It's actually quite quick to run. And so this turned out to be a very simple and effective way to do things, but not something, say, a machine learning purist would think was kosher. You know, it's somehow not really the right probabilistic way to properly model the problem. The other way is kind of better. So that was kind of the trade-off between the two. And even though this was nice and easy to do, we really wanted to be able to compare then, would this be as good as the other, or would the other be a better way to do things? So we did it both ways. And indeed, they led to significant improvements in the accuracy. And I won't go into all the details. And there were many details and many variations because that regularizer, for example, there's many ways to do that. And we did get significant improvements. The other thing Morella did was to really gather a very good and reliable data set. So that led to that was important in the process of improving the parameters. I haven't put up here the results of Doe et al. They also got improvements, but there were some caveats to their approach. They didn't use the thermodynamic data, so I haven't talked about fit with the thermodynamic data, and their results fit poorly with the thermodynamic data. Their initial paper didn't use a very large data set, so when we tested it on our big set, it didn't have very good accuracy. They then took our data set and trained their method on our data set, and they did get much better accuracy. But in the end, some of Morella's techniques ended up being the best anyway. One of the other things she did, which I won't talk about, is exploit known relationships between features in the feature set. So if you know, for example, that certain types of loops are likely to have similar energies, you can constrain the energy parameters using the regularizer to reflect that. And that can compensate for situations where you may have very poor data about some of the parameters because you're tying them to data that you have better information about. And overall, she got a 10.6% improvement on a huge data set in terms of accuracy. And a 10% improvement is pretty significant, I would say. Probably over the past 10 or 20 years, percentages of improvement between paper to paper would be on the order of 1 or 2 or 3%, whereas this was a big jump. So to finish this section about prediction, I just wanted to talk about some fun problems that you might want to think about. Here's a fun structure, which I call entwined hairpins. They're not kissing anymore. They're really entwined in a more substantive way. And I think it's a fun problem to ask whether you could predict the minimum free energy of such structures in n to the 5th time. Right now, you'd need n to the 6th. So far, I've also only talked about situations where you have a single strand. If you have more than one strand, the principles are awfully similar, really. I mean, base pairs are forming, loops are forming, and so on. But there's some very interesting differences that arise because if two sequences are identical, like if this purple one happened to be the same as the gray one over there, you have to account in your energy model for symmetry and other sort of effects that affect the energy. And when you do that, the problem just changes very slightly and subtly in a way that the algorithms don't work anymore. And what Dirks et al. Did was to figure out that if you wanted to calculate the partition function rather than the MFE structure, these additional terms canceled each other out in a very intriguing way that enables one to use some very nice results from group theory and plug those into the whole process to figure out the partition function, or the probability of base pairings. But their method does not work when you want to predict the minimum free energy structure. So I think this is a really fascinating problem from sort of a computational standpoint, algorithm standpoint, whether that can be done. And I haven't talked at all about folding pathways. I've just talked about the problem of predicting a static structure from a sequence. And there have been a lot of interesting work on folding pathways that I just won't get into. But again, I think there's a lot of opportunity there to do some interesting things. Those were open questions with regard to algorithms. Obviously, if we're at 70.6%, there's an awful lot of room for improvement on accuracy, too. And I think, again, there's fascinating problems in this area. There are specific features of the model that we kind of know aren't very good. In particular, the features pertaining to multi-loops are extremely simplistic. And an interesting question, and biologists know, like, oh, well, we should be modeling asymmetries in these loops, and we should be doing this, and yet the algorithms model is just a little simpler, and it doesn't quite do those things. So it's interesting to ask, would better features for multi-loops result in better accuracy? In order to figure that out, you'd have to write down a variation of the traditional Turner model. You'd have to redo the dynamic programming algorithm to calculate those energies properly, probably go from n cubed to n to the fourth if you were to do that. And then you'd have to plug all of that into Morella's machinery or something else to figure out better parameters. And then you could really test that hypothesis. And you might or might not do better, of course. You don't know. Could do a lot of work and not get so much better, or you might be really lucky and get 10% better. So who knows? So the next slide, I'll sort of get at that a little bit again. And then for pseudoknots, Morella has done some initial work, but I decided I wouldn't present it here, and I think it's, again, a huge area for improvement. Most of the work so far in improving parameters have been for pseudoknot-free structures. Okay, so one last thought on this is simply that the problem with trying this multi-loop idea that I put on the previous slide, let's try to improve the multi-loop model. Biologists know what the model should be. You know, we're smart enough, we could come up with a better dynamic programming algorithm, we could do this. But the problem is, it's a real pain. I mean, if you ask your student to do this, it's going to take them 6 to 12 months really to do it. And then in the end, you know, maybe it was a bad idea and it's no good. So, and then, okay, biologists will have another good idea. Or, you know, advisors are great to have ideas. Oh, why didn't we try a different multi-loop model? And then you go back into the lab and you program for 6 months and maybe it doesn't work, you know. So it's kind of it's just not that much fun, to be honest. So you could do it, but it just seems like a very painstaking way to make progress in this area. So I'm kind of curious. If we replaced true minimum free energy algorithms by heuristics, that wouldn't be guaranteed to give us minimum energy, but maybe guaranteed to give us pretty good or low energy structures. Those algorithms would typically use the energy model as a black box. So they would feed in a structure and say, give me the energy, and it would do cool stuff and lower the energy. And then it's really easy if you change your model, it's really easy. In 20 minutes, you could probably change your program to use the new model, because you just have to change the part that calculates the score. You're not changing details of complicated dynamic programming recurrences. Okay? So the question would be, if you replace the MFE by heuristics and you improved your parameters there, then could you feed those parameters back into the MFE and do better? So this would be a much more efficient way to explore a huge variety of models and maybe get much better results quicker, because ultimately you are going to use them for MFE, so you're going to be in good shape. And so, but I don't know if there's any theory or anything that might guide how you would do something like this in a rigorous way. And finally, I mean, all of what I've said is based on the premise that molecules actually do fold into their minimum free energy structures. And there's a lot of evidence, or some evidence to support that, but not every, you know, it's controversial, there's no proof. Probably for some molecules it's true and for others it isn't. So it's also important to look more broadly, in particular at the pathway that a molecule of structures a molecule would go through. Maybe it gets stuck in a local energy optimum or something, and try those algorithms and see how well you can do with those. So there's a lot of scope here, I think, for improvement. And since, you know, right now there's not so many people working on RNA compared to proteins, I think it's kind of a fun space to explore. Okay, so that's pretty much everything I wanted to say about predicting structure. And I thought I would finish up with some examples of why I also think programming sequences is a really fascinating area for research. Okay, so we've got DNA and RNA, and in a lab, the wet lab people can do pretty cool stuff now with DNA in particular. We can synthesize strands of DNA, any strand we want. We can form it into structures. These programs aren't great, but they're not bad. We can predict kind of what they're going to do. And so we can, in some sense, program them to do interesting things like molecular detection and things like that. So I think from a computer science standpoint, it's really interesting to think how we might go further with that. Humans have always taken materials from nature. You pick your favorite, anything, you probably have, our clothing, our furniture, everything, right? We take materials from nature and we build interesting things, very creative, new things. Well, if we worked with DNA in particular, which we can do quite a lot with DNA in the lab. We could start building things and doing things at the nanoscale, which is something we really can't do very well right now. So we have this amazing material that's totally digital that we can control, and we can use it to do interesting things at the nanoscale. And hopefully also eventually learn something about biology. But even if you didn't care about biology, right, just want to build a piece of furniture, you want whatever, then it's cool. So one way you could do this, so there's people in this field, there's just tons of ways you can do stuff with DNA. And I'm just going to give 2 examples, but they're just illustrative. You can do so many different things with it. So one is self-assembly. Self-assembly is a fundamental process in nature. Many structures we see in nature, like microtubules, flagella, and so on, are formed through self-assembly. And very abstractly, we can think of it as a process whereby tiles adhere to a growing assembly if certain glue strengths are sufficiently long. If you're a computational complexity person, you might think of weighing tiles and colors have to match, but glue strengths is kind of more close to a biological inspiration. So here's a tile, for example, that has glues that seem to fit with glues over here. And if maybe 2 different glues are needed for this thing to fit in and stick where it's supposed to stick. Okay. So this structure that I showed earlier is, in fact, a physical realization of a tile, which can be realized out of DNA at the nanoscale. And Eric Winfrey and his group asked if you could build interesting structures, programmable structures, by such tiles. So one thing that was a good test case was to build a Sierpinski triangle out of tiles. So you can do this, I won't go into the details, but a Sierpinski triangle, you can sort of grow one in a very natural way with 4 types of tiles. Here you can only see 1s and 0s. You actually would need sort of 2 types of 1s and 2 types of 0s. So that's a level of detail I won't go into. But the tiles will glue on in the right place, and a lovely, beautiful pattern like this will grow. And this is kind of interesting because it's not just a boring lattice. It's actually got aperiodic structure in some sense. And you can conceptually build it quite easily. So they use their little tiles. And you can see that these are images from atomic force microscopy that shows what they got. And they were able to distinguish between their 2 types of tiles in a certain way because of their design. And you can see that they kind of got something that looks kind of like Sierpinski triangles, although there are a lot of errors there too. Okay. So the point again is, you can have these ideas and you can really go into the lab and try them out and see if they'll work. And then you can go back as a computational person and ask a lot of interesting questions. How would you model and control errors that are arising in these situations? And you can ask even more basic questions, like if you were trying to build a particular shape or generate a particular pattern, how many tiles do you need to do that? And again, I won't go into the details, but there's some fascinating connections, for example, with Kolmogorov complexity and tile complexity that a student of Eric Winfrey's has discovered in his work. So I think there's really fun, interesting theoretical questions here. My second example is programming pathways as opposed to programming structures. we already saw an example where you could program pathways for an application of detecting small quantities of a molecule in a sample. Another application is something called a DNA walker. Here's a walker on the inside here. A piece of DNA, actually 2 pieces of connected DNA, that are going to walk along here and get to the end of this rail, guided by these hairpin structures and also aided by some fuel hairpins over there. So what's going to happen first is that the fuel hairpin, in this case, will get a toehold on the left hairpin, release the walker, so one of its feet is now flowing around in free air. And that foot is going to latch onto this toehold here. The word toehold is particularly appropriate for these feet. And it's going to disrupt this so that the walker finds itself in this position. And then the process is repeated. And, you know, if I had a few more hours last night, it could have gone a lot further. But since I didn't, I'll show it to you again, because it took me a long time to do that. Okay, so, right, you can build walkers, you can build pathways that help you detect tiny quantities of molecules. And walkers could be very useful for, you know, again, organizing matter in nanospace and moving stuff around in nanospace. And so there's many other things you could do. These 2 examples both use the same principle, which is metastable hairpins that are kinetically trapped in a sort of a particular state, and then a new thing gets introduced that releases them from that state and triggers a chain of reactions. And rather than just having these cool examples, you know, the people who've come up with these, particularly Niles Pierce at Caltech, is moving sort of further to the principles of how this would work and how you would abstractly model these kinds of systems and then compile these abstract designs into real DNA working systems. Again, I think there's a lot of scope for computational thinking to inform the process of moving forward with this agenda. So in closing, I hope I've convinced some of you at least that there are many compelling reasons both to predict and program structures and pathways that can be formed from DNA or RNA molecules. I think there's a lot of room for progress in prediction, in predicting pathways and improving the accuracy of current programs. I haven't said anything about tertiary structure. I think that's a whole unexplored domain too. And in terms of programming, I think there's huge scope right now to contribute to both models of and algorithms for computing or programming with biomolecules. So I just want to say thank you again, and thanks to various people who have contributed to this work, in particular Mirella Andronescu, who did the parameter estimation work. So any questions, I'll be happy to answer them. Thanks.
Other questions or comments?
Yes.
So you showed this example of the walker, I mean, and it looks really cute, but is that something
Thanks.
that actually works? I mean,
Yep.
Because there's a lot of random motion particles bumping into each other and so on. So you would be surprised to see it actually work in real life.
Yeah, so the way they test that something like this works is to attach a fluorophore to the end of one of these feet, or both of the feet, and also to the bottom of these hairpins. They attach things so that only when the 2 pieces come together that you get the fluorescent signal. And so they can sort of watch the signal move along the rail and see it happening. And they can, for example, use 2 colors, and then they can switch the colors on the hairpins and see that, you know, make sure that the colors are coming up in the right order, you know, and stuff like that. So yes, you can check those things. Yes?
In your PK3, you had an axis for accuracy. How are you measuring accuracy? Is it just a count of the...
So it's a little more sophisticated than the count. So we look at sensitivity and positive predictive value, and then we take the harmonic mean of those 2. But it's similar, you know. A simple way to think about accuracy is just the percentage of bases that you got correct. It's just a little bit more sophisticated than that.
For
Yes.
this worker pathways, is there any possible way to run it on the reverse direction on something like that? I mean, you can run.
That's a really interesting question.
Can this controlled under the reverse direction?
I think it should be, I mean, that's a really great question. I hadn't thought about it, but if you look at these things, you could design them. In fact, I kind of had a little piece sticking out on the top. You could maybe make that a little longer and have a different type of fuel that will take advantage of that and help run it backwards. It's conceivable. Do you understand what I'm kind of getting at? So I think it's conceivable that you could do it. These are pretty darn stable. So adding fuel that would disrupt these would be a little trickier than adding fuel that would have disrupted one of these hairpins, which is kind of in a metastable state, right? The hairpin, the top of it is destabilizing. So it's easier to disrupt this with fuel than it would be to disrupt this with fuel, but it's certainly conceivable that you could. Or, you know, what you could do is design your original piece of fuel so that when it latches on here, there's a little loop here in the middle that destabilizes it and that ultimately helps you knock it off. So it's a really interesting question. This is the kind of thing I think, you know, we want people to be thinking like this. How would you do it? Can you make it work?
If you can only control in one direction, you are kind of make something. Actually, if you can run on reverse both directions, you can try to keep some system running and express some kind of signal. Maybe you can do some computational.
Yeah, and if you could get that to work in the way that I'm sort of speculating you might be able to do it, you're freeing up your fuel, which is always a good thing, because then you can use it later on for something else, right? So, yeah, it's a very interesting question. Yeah, if you're interested in this kind of thing, you should come to the DNA computing meeting and talk to people there. It's a small community right now, and people love chatting about stuff like this. Or go to a wet lab and try it out. For sure. Seriously, that's the right thing to do. I wish I had the energy to do that, but I don't at the moment. Yes?
For that sensitivity that you were measuring for the base pair that you mentioned, did you look at the energy correlations, like this experiment? I know that you did it in your training, but for the final sensitivity, I'm just wondering how much does it correlate with the actual experimental energy value?
How sensitivity correlates with energy values.
After you did the training,
right.
So one measure is to count the number of base pairs that are correct.
Right.
Another way is to look at the predicted energy
yes.
that you have and compare that with the experimental energy.
Yes.
And I'm wondering, you know, whether did you look at that, or you know, is it a kind of measure that you should use for sensitivity instead of sensitivity?
Okay, so we did that, and I didn't report it here, and I don't think I could probably pull it up in a minute, but I don't. So anyway, we did it. So what you can do that for the thermodynamic data set, and there's over 1,000 structures in that set. These structures are very small and simple because people can get these thermodynamic measurements only for, you know, very, very short strands. And so, but we still had a lot of them, and for that set, we compared the predicted energies with the true energies, and we used a root mean square sort of sum to measure how well we were doing. And we found our numbers were, you know, low. I mean, 1 point something was the sum of the root mean square energies, which doesn't mean a whole lot out of context, but they were comparable to the original Turner numbers, about the same, and much lower than methods like Doe et al.'s method at Stanford that didn't use energies to inform the parameters.
So just roughly speaking, I just saw on one of these slides that you have minus 36 kilocalorie per mole. So in that range, is it usually very close? Like
yes,
1 or 2?
Exactly, yes. For those very simple structures. And we have no way of knowing for the longer structures. We have no energies for those.
Simplest structures, like how many base
pairs? The average was 17 base pairs, so between 10 and 30 probably. Yeah. So ours are certainly about as good as the original parameters in terms of what we can assess. Yeah. And that was very important to us in our work, that we respect those parameters. Yeah. And of course, if you really cared more, you could weight the term in our function higher so that those got even more weight. We kind of chose a value that seemed to work pretty well.
So the last question.
A more general question. Where do you see this research eventually leading? I know that I'm asking because you mentioned Adam in his paper, which he solved that, is it Hamiltonian? But the detractors of that paper said that the amount, at least for his setup, the amount of DNA we need to use to compute that would be an enormous amount. Do you see there being an application for computing hard problems using the research that?
No, I don't think we're going to be using this stuff to solve NP-hard problems. Yeah, but I think what we can do is program and control matter at the nanoscale. And I think there are reasons why we might want to do that. I mean, if you want to, organize tiny components on a chip and you want nanoscale precision, technologies like photolithography aren't going to work, right? They don't. The resolution of light isn't good enough to do that. But with this technology, maybe you could do more. It's speculative, but that's a possibility. So I think fundamentally being able to control matter at the nanoscale is a fascinating goal, and I'm sure it'll have applications, and I'm not too worried exactly what they'll be right now, but I'm pretty sure it won't be solving NP-hard problems. Yeah. And I think right now DNA is our best way of doing it. But in the long run, of course, when we understand proteins better, we might do it completely differently. Okay, well, thanks again very much.